Welcome back. In this series of lectures, we are going to be exploring the topic of muscle contraction, specifically skeletal muscle contraction. Remember, we're only talking about skeletal muscle right now. So, as we talked about last time, did you change your mindset? Did you change your words? When you were doing those couple of exercises I asked you to do, did you just sort of kind of do it and say, I know it good enough? Or did you change your mindset and ask yourself, is this really my best work? And then you went back and put a little bit more effort into it, okay? If you stop with the is it good enough, please turn off this video now, go back and redo those last couple of exercises until you know it's the best work you are capable of doing. And only then come on to learn about muscle contraction. Otherwise, you might be lost. Okay, so just a quick review of cellular respiration, because that's involved in this whole process of muscle contraction. So in cellular respiration, basically, the cells in the body are taking in sugar and oxygen, and they will be producing at the end of the reactions carbon dioxide, water, and energy. In a perfect world, around 36 ATPs. But in order to do this process, there are three small processes that are involved. So the first step is called glycolysis, which basically glyco, sugar, lysis, to break. So we're taking a 6-carbon sugar and we're breaking it down in the cytoplasm. This does not require any oxygen. To break down this sugar consumes a couple of ATPs, but we also produce ATPs. We'll start with a 6-carbon sugar and we end up with two 3-carbon molecules. And so we use 2 ATPs, but we produce 4 ATPs, so we have a net production of 2 ATPs at the end of glycolysis. Now, if there is oxygen present in that cell, then those 2 3-carbon molecules would then move into the mitochondria. And so when you look at the mitochondria, don't forget that it has an outer membrane and that inner membrane, and the inner membrane has some shelves, and then the mitochondria is filled up with its own solution. And that solution is called the mitochondrial matrix, and those infoldings of the inner membrane, those are called the cristae. So after glycolysis, when those 3-carbon molecules are entering the mitochondria, they're going into the mitochondrial matrix, and that's where the second step occurs, and that is known as the citric acid cycle, also known as the Krebs cycle. And a lot of things go on in that cycle. We don't have to know the minutiae details of that. This is not a chemistry class. The net result is we are going to be having some carrier proteins, NADH, FADH2, being produced. And those molecules then will enter the third step, which is the electron transport chain oxidative phosphorylation step. So whichever word you learned is fine here as well. Now this step also takes place in the mitochondria, but not in the matrix. It takes place on those inner membranes, in between the inner and the outer membrane, so on the mitochondrial cristae. And this is where we are going to have a massive production of ATP as well as water for a waste production. So hopefully at the end of this cellular respiration, we will have 36 ATP instead of just the 2 ATP net that were formed at the end of glycolysis. But remember, that would require oxygen. Now, if there's no oxygen, we do what's called anaerobic respiration, where that word anaerobic basically means no oxygen. And at the end of anaerobic respiration, we're starting with those same 2 3-carbon sugars that we ended up with at the end of glycolysis, but they can't go into the mitochondria for the Krebs cycle. So they're going to stay in the cytosol. And at the end, we can form one of two things. One option on your left is to form ethanol or alcohol. This is how you make beer in your bathtub or make wine, okay? If you think you're making beer or wine and you don't, it's because somehow oxygen got in your system, in your tubing or at a connection, because this can only occur in the absence of oxygen. All right, but that doesn't happen in our cells, so that's not the part we're interested in. Looking to the right side of the slide, this is what's happening in your cells, and that is the production of lactate or lactic acid. So with the lack of oxygen, we have a net production of 2 ATP, plus we're going to make lactic acid. And this is going to burn and not be good for your muscles. All right, switching topics, let's back up and look at the big picture, looking at the muscles. So for skeletal muscles to move, we are going to employ motor neurons, specifically somatic motor neurons. And then the axon of the motor neuron comes down, it branches into the axon terminal, at the end of which you're going to have your little synaptic knobs. And so each motor neuron is capable of stimulating multiple cells within the same named muscle. Okay, so even if there's two muscles, like the 4 quadriceps muscles all right next to each other, it's only going to be innervating one muscle. Okay. And each cell in that muscle can receive input from more than one motor neuron. So we talk about something called the motor unit. And what a motor unit is, it's the motor neuron plus all the muscle fibers, all the muscle cells that one motor neuron innervates. So if that axon comes down and branches into 10 axon terminals versus 100 or 1,000 axon terminals, you can see how large the motor units can become. Now, all of these cells being innervated in that one motor unit, in other words, that one neuron, all the cells that it's innervating, they are going to contract in unison. So in other words, they're going to behave just like one unit, a single functional unit. And those cells are not all smushed right next to each other. They are kind of widespread throughout that muscle. So when that one neuron contracts, it's giving a widespread contraction that seems weak because it's all over the place, as opposed to a really strong contraction in just one little area. So looking at the picture on the bottom right, here's the advantage of this type of system. This is showing 3 different motor units. And they have 3 different color cell bodies for the 3 neurons, and those are related to the same 3 color muscle cells. So for instance, the pink motor unit, what's motor unit number 2, goes to the pink cells. So you can see it's innervating 3 cells in this diagram. The motor unit number 3, which is peachy, is only going to the peach cells, and there's only showing 2 cells there. And then the yellower, whiter one, motor unit number 1, is also innervating 3 cells. So what happens is this is going, all of these are in the same muscle, but all of these motor units are not contracting at the exact same time. So they're doing what I like to call shift work. Okay, we got a day shift, an evening shift, a night shift. Okay, well, it's not 8-hour shifts. Okay. But what happens is, say for instance, motor unit 1 is going to contract those 3 muscles, and meanwhile the other 2 on rest, and then motor unit 2 is going to come in while those are contracting, and it's going to relax, and then maybe 3 will come in while 2 is still contracting and 1 is still relaxing. Okay, so basically these motor units are going to be rotating when they're contracting and when they're relaxing, because they get fatigued quite quickly, and so they can't sustain a contraction. However, the fact that we have motor units means that muscle as a whole can have a longer muscle contraction. And so that's the advantage of the motor units. All right. So the next thing we want to learn about motor units is the size of them, because on the previous picture, 2 of them showed 3 muscles and 1 of them showed 2 muscles, and neither of those are really realistic. Okay. But we do talk about, for every axon, how many muscle cells are being innervated. So this gives us a ratio. The lower the ratio, the lower number of cells being contracted per axon, the finer the control is. So I guess it's the higher the ratio. Anyway, let me give it to you in numbers so you understand it. Typically, it's about 1 motor neuron going to 150 cells, not the 3 that I showed, but 150. In places where we want super fine muscle control, that 150 number is going to be a much lower. Okay. So this would be what's called a small motor unit, where fine control is needed. And a great example of that is in our muscles of our eyes, because we don't want bunches of cells contracting. So we're looking here, and then all of a sudden we're only looking there. We want to be able to look every single place, okay? And we'll have some small motor units in our hands for writing as well. In contrast to small motor unit, for instance, the eye muscles, 23 cells contracting per neuron. A large motor unit comes into play where strength is more important. An example of this would be like in your quadriceps or in your hamstrings or in your buttock muscles, where upwards of 1,000 cells are going to be innervated per neuron, giving you a lot of muscles contracting at once, giving real power behind that. All right. Another concept with the motor unit is that it follows what's called the all or none principle. When that impulse comes down the axon, it's going to go to all branches of the axon terminals. So it's going to go to every single muscle fiber in that motor unit. And so all of those will either contract or none of them will, okay? And all of them will contract fully. So it's not as if, okay, we have a really weak action potential going down, so let's only have like 30% of the cells contract. That can't happen. Or let's have them all contract, but let's have them contract like this instead of like this. Okay. That's not going to happen. All right. Continuing on. Do you remember the neuron we already learned about? Remember, we had the cell body where most of the organelles were. Attached to that, we had dendrites bringing all the incoming messages. And then we had a single axon with the outgoing messages, which then split into the axon terminals. The ends of each was a terminal bouton. Okay, just want to refresh your memory because the terminal bouton is going to come into play again with muscle contraction. And that's because of a structure known as the neuromuscular junction. So look at this word. What 2 cells are involved? Yeah, a neuron and a muscle cell. Sometimes anatomy and physiology are that easy. All right, so the neuromuscular junction is the functional side of communication between a neuron and a muscle cell. Now, they don't actually touch each other. Okay. So let's go through the anatomy of a neuromuscular junction. First, we have the neuron, this yellow cell. From the neuron, we had the axon, which had the axon terminals, which is what you see up at the very top of the picture, and then this bulbous part at the end, that's the terminal bouton. In the terminal bouton, we have those synaptic vesicles filled with their neurotransmitter. In this case, it's going to be acetylcholine. The space between the neuron and the muscle is known as the synaptic cleft. The muscle cell, the plasma membrane of the muscle cell is called the sarcolemma. Inside the muscle cell, we're going to have lots of mitochondria so we can make lots of ATP, because remember, cellular respiration, if we have the mitochondria, we have the oxygen, we can make 36 ATPs instead of 2 ATPs. This is also where we're going to have our myofibrils, our myofilaments, those proteins, myosin with the overlapping actin. And also don't forget our friend, the T-tubule, which is the extension of the sarcolemma going across, forming part of that triad, which we will see shortly. So let me talk about acetylcholine just for a minute. This is the structure of acetylcholine. You don't need to know it. I just put it there just so in case you wondered what it looked like. Any synapse that uses acetylcholine as the neurotransmitter, such as this neuromuscular junction we're talking about, is called a cholinergic synapse because of the choline in acetylcholine, a cholinergic synapse. So how do we form acetylcholine? Well, acetyl-CoA is one of the two things. Remember acetyl-CoA? That was one of the little things that enters the mitochondria for the Krebs cycle. Acetyl-CoA will bind to choline, jumps off the CoA part, and that forms acetylcholine. So acetylcholine can be both excitatory and inhibitory. For the neuromuscular junction and skeletal muscle, which is what we are talking about, it's going to be excitatory. Okay, so it's excitatory for those postsynaptic muscle cells. But in other postsynaptic cells, it can be inhibitory. For instance, this is what can happen in skeletal muscle. Another structure I want to introduce you to is something called the motor end plate. And the motor end plate is a region, it's an area on the sarcolemma. And it's always opposite where these terminal boutons are located, because it's in the motor end plate where we find millions of acetylcholine receptors. And this makes sense because we would not want to have, for instance, all of our acetylcholine receptors up in Sacramento if our terminal bouton was down in San Diego. That wouldn't be very useful. So the motor end plate is going to be opposite the terminal bouton. So looking at the motor end plate a little closer, what you will notice is the sarcolemma at the area of the motor end plate has all these infoldings. And these infoldings are just to increase the surface area so it can contain even more acetylcholine receptors. And each of these acetylcholine receptors will bind to acetylcholine. So if it's only binding one acetylcholine, that receptor still can't do anything. It will require 2 acetylcholines to bind to it for that receptor to do its job. And so we call this acetylcholine receptor a ligand-gated channel because it requires some kind of molecule to bind to it, a ligand-gated channel, acetylcholine being a ligand. Okay. So when the 2 acetylcholines bind to the acetylcholine or ligand-gated acetylcholine channel, then what happens, the receptor, excuse me, then a channel opens up in that receptor, and that channel will allow sodium to enter the cell, thereby depolarizing, but it also will allow potassium to exit the cell. So the same channel on the same receptor allows both diffusion inward of sodium as well as a slower diffusion outward of potassium. And that brings me to an enzyme known as acetylcholine esterase. So I want to point out this abbreviation. The abbreviation for acetylcholine, you do need to know the word acetylcholine, but the abbreviation is a capital A followed by a capital C and then a lowercase h. So acetylcholine esterase, the enzyme, after you've written it a couple of times, you take the acetylcholine abbreviation and you put a capital E after it. Okay. So Once acetylcholine has bound to, two of them have bound to the cholinergic receptor and the channel has opened, almost immediately they detach and they're back in the synaptic cleft. And acetylcholine esterase lives in the synaptic cleft. And what the acetylcholine esterase molecule does right here in the synaptic cleft, the acetylcholine is going to be met up with the acetylcholine esterase, and it's going to break it down into two components. Acetate, not back to acetyl-CoA, because remember, it got rid of the CoA part. So it's going to break it down to acetate and choline. The choline, by endocytosis, will be retaken up into the presynaptic terminal bouton in a little vesicle, where once again, it can meet up with some more acetyl-CoA and form more neurotransmitter, acetylcholine, sitting in a synaptic vesicle. Meanwhile, the acetate that's there in the synaptic vesicle is just diffusing away to areas of lower concentration. So here is a picture at the bottom, which is kind of putting everything together that we've talked about so far. So make sure you stop the video right here and take a look at it and talk yourself through everything we have talked about on this section before we start with our muscles contracting. Thank you, and I will see you again shortly.